Hello, everyone. Today we embark on a detailed examination of the regulatory mechanisms governing glycolysis and glucogenesis, with a particular emphasis on the pivotal role of aldolase gene expression. These pathways represent fundamental pillars of cellular metabolism, one catabolizing glucose to harness energy, and the other anabolically synthesizing it from non carbohydrate precursors. Aldolase serves as a critical enzymatic nexus, its isoforms and regulatory expression patterns adapting to diverse physiological contexts. Throughout this presentation, we'll elucidate their interconnections, allodase's bidirectional functionality, the isoform variation, and the transcriptional orchestration of aldolase B. By providing insights into metabolic homeostasis and pathophysiological implications. Our learning objectives. These are going to set the framework for our discourse. First, to comprehensively explain glycolysis and glucogenesis, highlighting their reciprocal interrelations in energy metabolism. Second is to describe aldolase's mechanistic actions in both the forward and reverse directions. Third, we're going to compare the three aldolase isoenzymes, A, B, and C, as well as their tissue-specific expression profiles. And finally, to discuss the transcriptional regulation of aldolase B by key factors such as HNF-1, HNF-3, C over EBP, and DBP. I know, but don't worry, we'll get into all of it. Lastly, we're going to be underscoring how these elements integrate hormonal and nutritional cues for hepatic function. Glycolysis stands as a cornerstone of cellular energies, transforming one molecule of glucose into two pyruvates while netting 2 ATP and 2 NADH. This systolic process operates in nearly all eukaryotic cells, bifurcates into an energy investment phase and a payoff phase. In the investment phase, hexokinase and phosphorylates glucose to glucose 6-phosphate, expending 1 ATP, followed by isomerization to fructose 6-phosphate and phosphofructose kinase 1s. That's the commitment step. Adding another ATP to form fructose 1, 6-biphosphate, a regulatory bottleneck responsive to allosteric modulations like AMP and citrate. Aldolase then cleaves into dihoxyl phosphate and glyceraldehyde 3-phosphate, with triose phosphate isomers enduring both proceed. The payoff phase recoups investments. That's going to be our glyceride 3-phosphate. Dehydrogenase generates the NADH, and the 1 and 3 biphosphoreglycerate. The phosphoglycerate kinase yields ATP and 3-phosphoglycerate. That is a hard word to say quick 3 times. Now, subsequent mutase enolase and pyruvate kinase, those steps are going to produce another ATP per triose, culminating in pyruvate. So this pathway not only fuels anaerobic conditions, but it also supplies intermediates for biosynthesis and its regulation fine-tuned into cellular energy status. To encapsulate, glycolysis unfolds in the cytoplasm universally, converting glucose to 2 pyruvates with a net gain of 2 ATP and 2 NADH, as we just saw. Regulatory enzymes, the hexokinase, PFK-1, and the pyruvate kinase, they're going to ensure that the responsiveness to the metabolic demands, prevent any overreproduction when energy is abundant. So, this net yield underscores its efficiency as an ancient conserved pathway, integral to both intermediate or immediate energy as well as linkage to aerobic respiration via the citric acid cycle. Gluconeogenesis, the biosynthetic counterpart to glycolysis, synthesizes glucose from precursors like lactate, glycerol, and amino acids, predominantly in the liver and the renal cortex to sustain euglycemia during fasting or prolonged exercises. The intensive pathway demands 4 ATPs and 2 GTPs per glucose molecule. Key bypass enzymes circumvent glycolysis' irreversible steps. The pyruvate carboxylase, biotin dependent. It converts pyruvate to oxalate in the mitochondria using ATP. The PEPCK, or phosphoenolpyruvate carboxygenase, then forms phosphoenolpyruvate with the GTP in the cytosol. Fructose 1, 6-bisphosphatase, hydrolyzes the fructose 1 and the 6-biophosphate to then become fructose 6-phosphate and glucose 6-phosphatase, which liberates the glucose. These adaptations ensure unidirectional flow, highlighting gluconeogenesis' role in the interorgan metabolic cooperation, such as the Cori cycle with muscle lactate. Now, focusing on organ-specific contributions, gluconeogenesis averts futile cycling through reciprocal regulations with glycolysis. Those are modulated by ATP, AMP, citrate, and the pivotal allosteric regulator, fructose 2, 6-biophosphate, which is going to be the, the main party that stimulates the PFK-1 while inhibiting fructose 1, 6-biophosphatase. So in the liver, the kidney cortex, this pathway is going to uphold systemic glucose homeostasis, with hormonal inputs like glucagon enhancing the PEPCK expression via the key CAMP, that's countering the insulin suppressive effect. This organ centric orchestration exemplifies adaptive metabolism. It's crucial for preventing hypoglycemia in catabolic states. Aldolase facilitates a reversible aldoid reaction central to both pathways. In glycolysis, it cleaves fructose 1, 6-biphosphate into dihydroxic acetone phosphate, or DHAP for short, and our G3P, or glyceraldehyde 3 phosphate, enabling triose phosphate progression for ATP generation. Conversely, in gluconeogenesis, it condenses DHAP and G3P back to fructose 1, 6-biophosphate. The class 1 aldolase mechanism involves a SCIF-based intermediate, where a conserved lysine residue forms an imine with the substrate's carbonyl, stabilizing the enolate for carbon-carbon bond manipulation. This bidirectional capability, operating near equilibrium, underscores aldolase's versatility in metabolic flux control that adapts to the cellular energy demands without requiring bypass enzymes. Let's talk about aldolase. Now, it exists in 3 isoforms, each encoded by distinct genes and optimized for tissue-specific roles. Aldolase A, or ALDOA, predominates in skeletal muscle and erythrocytes. supporting high glycolytic rates for rapid ATP production. Aldolase B, ALDOB, which is found in the liver, kidney, and intestines, facilitates gluconeogenesis and fructose metabolism with enhanced affinity for fructose 1 phosphate. Aldolase C, or ALDOC, found in the brain, sustains neuronal energy needs. These tetrametric enzymes, sharing about 70% sequence identity, exhibit kinetic differences. So, Aldolase B, they have a lower activity suits for the hepatic bidirectional flux. The mechanism reiterated here relies on the shift base for substrate binding, exemplifying evolutionary conservation in metabolic efficiency. Regarding Aldolase B expression control, we'll explore its transcription regulations next. Building on isoform diversity, Aldolase A localizes to glycolytic tissues like muscles for bursts of energy, whereas B anchors the gluconogenic organs like the liver for sustained glucose output, and C in neutral tissues for a constant supply. So kinetically, A boasts high Vmax for the F1 6BP cleavage, which is ideal for anaerobic bursts, while B's broader substrate specifically includes fructose 1 phosphate, which is vital for dietary sugar handling. Functionally, this specialization ensures metabolic compartmentalization, with implications for disorders. An example, Aldolase B mutations in hereditary fructose intolerance disrupts liver functions. Significantly, these patterns inform targeted therapies in metabolic syndromes like diabetes, where isoforms dysregulation contributes to the hyperglycemia. Transitioning to regulation, HNF1 and HNF3 confer liver specific activation through the HNF3, often competes antagonistically. C over EBP responds to fasting via glucagon inducing signaling. The DBP imparts circadian rhythms, integrating temporal cues into expression. In glycolysis regulation, Aldolase's reversible steps integrate seamlessly. Modulated indirectly through upstream effectors like the PFK1, its roles extend to gluconeogenesis, where condensation supports glucose synthesis. Clinically, ALDOB mutations precipitate hereditary fructose intolerance, wherein fructose 1 phosphate accumulates, sequestering inorganic phosphate and inhibiting glycogenesis. This leads to severe hypoglycemia, gastrointestinal distress, and potential hepatic failure upon fructose ingestion. This exemplifies how Aldolase dysregulation cascades into systemic metabolic perturbations. Aldolase's function in gluconeogenesis is indispensable, enabling the condensation step amid energy expenditure. AMPK, an energy sensor, indirectly enhances Aldolase activity by promoting catabolic shifts into low ATP states. Hormonally, glucagon upregulates glucogenic genes, allowing ALDOB via CAMP, while insulin represses them, illustrating cellular adaptability to nutritional status. This interplay ensures metabolic resilience with Aldolase is key to an adaptive node. Now, here we have our summary. Let me just say I am so proud of you for trudging through our examination of glycolysis and gluconeogenesis. Personally, I look at this information and, the vocabulary alone is intimidation. So, just to ensure that you grasp the concepts that we went over today, I'm going to go ahead and speak plainly here. Now, our bodies are like smart factories that can make energy when we need it and save energy when we don't. The two big helpers make this happen. First, glycolysis. They break sugar or glucose apart to make energy. Second helper is gluconeogenesis, who builds sugar back up when we run low. Now, when we look at glycolysis, they're breaking sugar for energy. You can think of glucose as a candy bar for your cells, and glycolysis comes in and cuts that candy bar into smaller pieces, and we call them pyruvate. And that is how we make our ATP. ATP can be thought of as like little tiny batteries that power our body. And keep in mind that this is happening in every single cell all the time, especially if you're out running or playing or having, even if you have to think too hard. The process spends a little bit of energy first, but then it earns back even more. An example can be like you pay $2, but you make $4 back. Okay. Now, our second helper, gluconeogenesis, they are building sugar when you're hungry. So, when you haven't eaten or you're fasting, your body can't get sugar from the food. So, what it does is your liver and your kidneys, they use leftover bits, whether it comes from fats or amino acid or even lactic acid. They figure it out and they make new glucose. It's basically like recycling. If you had old building blocks, you're going to use those to build a brand new candy bar. Now, this costs energy to do. However, it keeps your brain and your muscles working when you're sleeping or if, like I said, fasting. Which brings us to our aldolase, the super switch enzyme. In both of our helpers, as they complete their jobs, there's one special enzyme called aldolase. It's like a switch that can work forward when we're breaking sugar down, or it can work backwards when we're building sugar back up. It cuts a sugar molecule in half and can also glue those halves back together when you need them. Aldolase is made in a different flavor from the rest of body parts. As we went over, aldolase A is in muscles for those fast energy spurts that we may need. Aldolase B, those are found in the liver to help save and make sugar. And aldolase C is in the brain to help you think. But how does the body control aldolase? Well, your body has these tiny switches in your DNA, and that tells them when to make more or less aldolase. These switches respond to hormones like insulin and glucagon, and even your daily rhythm, that's to say the daytime or the nighttime. When you eat, your body slows down gluconeogenesis. When you're fasting or sleeping, it speeds up thanks to aldolase B in your liver. Now, when things go wrong, if the aldolase B gene has a mistake, the liver can't handle sugar from the fruit correctly. This causes hereditary fructose intolerance, which can make a person very, very sick if they eat too much sugar. Now, scientists have been studying these enzyme problems to help design new medications and better diets for our citizens. But overall, the big picture, just so we all understand and are on the same page, when we talk about glycolysis, it means we're making energy from sugar. Gluconeogenesis, saving energy by rebuilding sugar. And our aldolase is the tool that we use that can do both. Your body uses teamwork to keep blood sugar steady help you play, think, and grow even while you're sleeping. It's like having a power plant and a savings bank inside every single cell, working together every second of your life. I hope that was a better layout of all of the information that we did go over today. Here we have our references. These references underpin our discussion, drawing from authoritative sources of biochemistry and metabolic regulation. I would like to go ahead and thank you all for your attention. If you have any type of questions, comments, or concerns, go ahead and feel free to reach out to me. I welcome any questions on this intricate pathways. Be blessed and have a great rest of your day. Aloha.